This is the January 2012 paper. It's the BY1 paper, and we're looking at question number 8, which is the essay question. We're going to look in this video at the part A question, which is about enzymes, okay, and in particular it's about the use of immobilized enzymes. Okay, so it's a 10 marker as normal with these essay questions. This first essay then is a reasonable one. Okay, as long as you've learned all the factors about immobilized enzymes and of course the biosensor topics as well, you should be reasonably okay with this. The thing to remember, of course, is you've got to remember how enzymes are immobilized. So you should be able to state how immobilization occurs. I'll pull up the notes that I've included in the app in a moment to show you where the relevant information is relating to this question. Okay, so the other thing you need to remember, of course, is the advantages of using immobilized enzymes. That would be sort of important for this essay. So things like you can reuse these immobilized enzymes. Hopefully you know about producing lactose-free milk. Any sort of industrial process, you can actually reuse these immobilized enzymes because they're immobilized in an inert matrix. Of course, they don't enter the product. So another advantage is that they are or they don't contaminate the product. Okay, they can be reused. They can tolerate a greater temperature and even a greater range of temperatures as well. So they're more sort of thermostable because of their entrapment in things like alginate beads. The other one, of course, is you can use several enzymes together to give sort of different products if you were doing an industrial process. So there's lots of advantages that you can state in this essay that you'd have by using immobilized enzymes. So that would cover quite a range of the biological principles there in immobilized enzymes. But you are also asked to talk about biosensors. Okay. Now, again, you should be able to state why biosensors are important. There's two types of biosensors. There's this clinistick example, which is a qualitative biosensor, if you remember. It's one where you just get a color change. The step up from that is the digital biosensor, which is the major one that you need to know for your exam. So you can mention a couple of advantages of using biosensors. For example, they're accurate, they're highly specific. Because they use enzymes, it means that they will only detect a specific substance. Okay. They're very sensitive, which means they can detect low concentrations of a substance. Okay. They're used, as it says in the question, they can be used to detect blood sugar concentration. Okay, and of course that's used by diabetics because they can't regulate their sugar concentrations. Okay. The other thing that you need to put into this essay is the functioning of particularly a digital biosensor. Okay, you will get a mark for mentioning the clinisticks and saying about qualitative biosensors, but the major emphasis here is on quantitative biosensors, which are the digital ones. So you should be able to describe the functioning of each part of a biosensor, for example, the transducer, the semi-permeable membrane. You need to know that in a biosensor that detects blood glucose levels, you should be able to name the enzyme. The enzyme, of course, is glucose oxidase. Okay, so if I just pull up the notes that accompany the app, and I can show you where all this relevant information can be found. So here's the notes starting with immobilized enzymes. We've got a definition there. It's important that you remember the definition. Inert matrix. Okay, an immobilized enzyme is an enzyme that is attached or trapped in or on an inert matrix. Lots of people forget to mention inert matrix. It is important. Okay. Moving on down then, if I go to the next page, we've got various methods of immobilizing enzymes. Okay, the most obvious one, and the one that's always in mark schemes, is to sort of trap the enzymes in alginate beads. Okay, so there's various information there. The next one here is sort of a method for immobilizing enzymes in alginate. Okay. This slide here looks at the qualitative biosensor, which is the CliniStick. You just get a color change. Okay, so the next slide we'll look at the digital biosensors. Okay, and further on we've got, of course, pictures here which show the general arrangement of the components of a biosensor. So you should be able to talk about the immobilized enzyme, the selectively permeable membrane. Okay, here, immobilized enzyme there. The transducer is important. Okay, remember the transducer will absorb the products from the immobilized enzyme, and it'll convert that product, which is a biological molecule, it'll convert it into an electrical signal. Okay, and that's very important to explain in exams, that this transducer can convert a biological signal into an electrical one. So that's the general features of a biosensor there. Later on then I talk about the functions of each of these components. Okay, remember that the selectively permeable membrane only allows the substrate to enter the biosensor. It prevents large molecules like blood cells and large proteins from entering. So that's the function in there of the biosensor. Okay. Which is all listed in these notes. There's advantages again of biosensors. So if you look at the notes, all the relevant points are found that you can actually put into this this essay. Okay, so I've started writing in an answer now for this enzyme question. I've just made general comments now about how to trap the enzymes in the inert matrix. I've mentioned the alginate beads. I've stated that they're industrial as well as medical uses. I've started mentioning a bit about the industrial use and forming lactose-free milk. I've then, that has allowed me then to enter and state some of the advantages of using immobilized enzymes. So as you can see there, I've mentioned that they can be reused. All right, they do not contaminate the product. Okay, I've said that they have greater stability, so they can tolerate much higher temperatures or pH. All right, so all of that now would be worth quite a few marks in the essay. In medical uses then, I've said that we use the biosensors. Okay, I've then started to talk about the properties of the biosensor. So I've said that they are very sensitive in that they can detect low concentrations of a particular substance. All right, I've said that they are very accurate and they are specific. Okay, I've also then said they can be used to measure glucose concentration, and I've mentioned that the enzyme here, glucose oxidase, can measure glucose concentration. All right. I've also said then that the biosensors can be used by diabetics. That would also get you the mark. So all that's left really to do in this question is to talk about the functioning of a digital biosensor, look at the different parts of the biosensor and explain what they do. You could draw a diagram here, but be warned, any diagram that's drawn must be fully annotated. And what I mean by annotate is actually give a description. Annotate does not mean label. The diagram must be labelled as well, but to annotate it you need to give a description of each of the components that you draw. Now I'm not going to draw the biosensor in, I'm just going to describe the functioning of each part of the biosensor. So please remember, if you draw diagrams you must annotate them, which means fully describe what you have drawn. So I've typed in the last part of the essay. Here I'm just describing the important components of the biosensor. So I've mentioned the selectively permeable membrane initially, okay, which is there. I've stated that this membrane will only allow the substrate to enter and prevent large substances like blood cells from entering. It is important, I think, because of the question is asking about glucose measurement, that you say that the substrate is glucose. Alright, I think that's very important to put in essays as well as short answer questions on immobilised enzymes. I've then said that the glucose will be converted into a product by the glucose oxidase, which is the enzyme in the biosensor. The product will then be absorbed into the transducer. Alright, and then I've described the function of the transducer. It will convert the biological signal. So the biological signal is actually the product formed by the enzyme reaction in the biosensor. So this biological signal then is converted into an electrical signal, and that will get you a mark as well. And lastly, and this is also mentioned in the notes I've produced, the size of the electrical signal, okay, is directly proportional to the concentration of the product. All right, so that's quite important to mention as well, and that is explained in the notes. And lastly then, I've just said that the electrical signal is then displayed as a digital readout. So that is a good answer there. It covers everything that the examiner wants. It covers everything that you should have learned in your A-level. And that should get you then 10 marks.